So this video is all on the process of DNA replication. This is going to focus on mostly level 2s and some level 3 objectives regarding replication. So if we do a quick review, you'll recognize this picture here in the center as being the cell cycle. So if you recall, DNA is replicated during the S phase of the cell cycle, and the reason it's called the S phase is because it stands for DNA synthesis. To synthesis means to make, create, or to build, and that's exactly what we're going to do. So DNA replication is a multi-step process that happens with many different reactions. So because I have many different reactions taking place, I'm going to have many enzymes involved in these different reactions. So we're going to take a look at the different steps and the different enzymes that are involved here. Alright, so let's take a look at this model over here. What we've got here is I've got my sugar phosphate backbone on the sides. So my sugar and phosphates, you'll see that on both sides here. And I have my nitrogenous bases in the center. So I've got my A's and T's and G's and C's. So step one of DNA replication is we're going to pick a location that we're going to start to replicate. So enzymes are going to come in and they're going to bind to the double helix DNA in the location that we would like to replicate. Now, if we only start at one place within the DNA and then had to replicate the entire thing, it would take forever. So what ends up happening is that we pick many different locations along the same strand where this is happening at the same time. So the place where this is going to happen is called a replication fork. And we'll see why it's called a fork in just a short bit. Okay, so step two, we're looking at our first enzyme coming in, and that enzyme is helicase. And helicase ends in ASE, so I know it's an enzyme because of the enzyme, or because of the ending, and it's going to come in and it's going to unwind the double helix. Okay, so I'm just going to abbreviate it. And the way it can do that is that it has to break the hydrogen bonds that are holding the bases together. So if I look in the model, between A and T, there was a hydrogen bond that was connecting them. Well, helicase comes in and it breaks the hydrogen bonds. Same thing here, the hydrogen bond was broken, so helicase is going to move down the double helix, and as it comes through, it's going to break all of these bonds. And so basically it's going to unwind it or open it up like a zipper. So after helicase continues to come through, so here's our helicase enzyme, and it continues to go through and basically break all of those hydrogen bonds. So it keeps moving down. We're going to see that we now have these two strands over here, which now are unpaired. And if they're unpaired, they're unhappy. So step three is we're going to form the new strands. So our next enzyme that's going to come into play is called DNA polymerase. And DNA polymerase is responsible for bringing in new bases to pair up with the unpaired bases on the original strands. So the enzyme would literally be looking around the nucleus and say, all right, if I take a look at this base up here, they'd say, well, we need a thymine. So DNA polymerase would grab a thymine, it would bring it on in, and then it would bring it on. Next to it, the bonds, the hydrogen bond would then form in between the A and the T. Then another DNA polymerase would come on in and bring an A. Then another DNA polymerase would come on in, and that would bring a cytosine. And then another DNA polymerase would bring in a guanine, and then bring in another guanine. So the DNA polymerase is helping to kind of bring in these new bases and form the new strand. Now the same thing would be happening on the other side, but on this side it's going to go in the opposite direction. We'll see if you guys remember why. So on the other side, This would bring in a cytosine, then a cytosine, then a guanine, then a thymine, then an adenine. And then this one is going to grow in the opposite direction. So basically, DNA polymerase has two jobs. One job is to bring in those new bases to form the new strands. But it has a second role, and its second job is to proofread. And it is so important that those bases, or that the right bases, are brought in, because if there is a change in the DNA, then we end up with a mutation. And that we do not want. So basically it's going to go on through and it's going to double check and say, all right, do we have the right base pairs? So it will go through and literally double check all the base pairings and make sure that they are all paired up correctly. So the last part of forming the new strands is we have one final enzyme that comes in, and it's called DNA ligase. Once again, it's an enzyme, it ends in ase, and ligase is going to come in and kind of glue all of the kind of fragments and pieces all together and kind of solidify the backbone. So in our backbone, we want to make sure that any gaps that we might have when we brought in those new bases, that we're going to solidify and glue all those gaps back together in the backbone. Now if you recall, what connects the sugars and the phosphates together in the backbone would be covalent bonds. So the covalent bonds are in the backbone, while the hydrogen bonds are what's connecting the two bases together in the middle of the DNA molecule. So by the time you are all done, we know that we're going to end up with two identical strands of DNA. If I look at the left side and the right side, they should be exactly the same. So if I look at this side here and I look at this side here, they are identical, and that's the whole point of DNA replication, is to create two identical strands. However, what we call this is a semi-conservative model. Semi means halfway, conserve means to save. And what they're saying is half of it is saved. Basically, I'm ending up with half of it new and half of it original. So if you look in the picture, the black strands were the original strands. So if I look at my picture here, like literally half of it is the black strand, and then half of it is the orange strand, or the new strand. Okay, and the same thing is true on this side. So they call it semi-conservative. Half of it is new, and half of it was the original DNA that we started with.